All right, so chapter 17, this one should be pretty quick just because there's just not a lot of information to this, and that's why there's less questions on the quiz and everything like that. So we'll run through it. I do think it's actually pretty important, though, as far as medical field understanding of microbiology. So I would definitely try to take the time to understand the concepts here. So chapter 17 is dealing with diagnosis and diagnostics. So the tools that we would use to diagnose an infection. We can basically break down the types of tests that we would be doing into phenotypic, immunologic, and genetic. So phenotypic, that's going based on symptoms that we might see, basically anything that you would observe up front, measuring the temperature, the fever of the patient, and all that stuff you can observe up front for the patient and record that way. Immunologic, now we're going to be doing tests that have to deal with the immune response or identifying a pathogen that's in someone's body using antibodies that we know of. And then we have the genetic tests. This is what it sounds like. I don't know. It's like PCR and stuff like that. It's literally looking at the genome of the potential pathogen and trying to determine if it is, in fact, the thing we think it is. So how do we, approach all of this? Well, the first thing you're going to do is begin with the patient, and we're going to look at signs and symptoms. Signs and symptoms, basically a sign is anything that you can record, like a fever, for example. Swollen lymph nodes, you can actually palpate that. Vomiting itself, yes, that counts, but symptoms, that would be like pain, because you can't even tell really pain, you can't prove it, right? You just to go based on the patient's words, like what they're saying, what they're telling you, or fatigue, nausea. You can't actually measure these things. That brings us back to that concept of the syndrome, which I think we already brought up earlier, but a syndrome is essentially a set of signs and symptoms and other factors that we can associate with disease, but it can be caused by a number of different causes, like meningitis. We can have viral, we can have fungal, and we can have bacterial, and there's all different bacteria and everything that can cause these things. That's just one example. So that's a syndrome. All right, so you can assess all of these things. These are kind of phenotypic types of approaches. Then we can collect the appropriate specimens and analyze. So if the person has a sore throat, you might swab the throat. The person's coughing, you might collect their sputum, which is just they cough up and spit it into a little cup. And it might sound gross, but it's actually a pretty effective way for diagnosing respiratory infections, especially tuberculosis, which is actually a pretty common infection. So we'll do that, and then we can analyze these directly using a microscope or immunologic methods or genetic methods, just directly onto the sample itself, or we might want to culture it. If we can't get the answer that we want, or it's like, too much of a mix, like stool sample, there's too much going on in there. You can't always look at it directly unless there's special tests made for that specifically. So you might want to culture it and isolate it first. So you would isolate it and identify it using whatever variety of tests that were available. And that's kind of what we're doing in lab right now, right? We're using our stains and eventually our physiological tests in order to determine what our unknown bacteria are for each group. So that's the idea with that. Presumptive data is basically preliminary categorization. So we could say, oh, well, this is definitely gram-negative, or this is, some sort of staphylococcus. And then confirmatory is going to pinpoint it to the specific identity of the organism, so the species and even the strain. Cultivation, that's just a term referring to, growing up your sample on isolation media. You could use differential media to choose things. So differential or even selective media. So differential lets you see like the color changes. And so you might, if it grows on this particular media and it makes pink colonies, and then you have other ones that didn't turn any color, and you think that your pathogen should be pink on that media, that's a way to distinguish. Or even like growing it on actual differential media and just recording, what color it gave you. So you did the throat swab and you put it on a differential media. I don't know, like you wouldn't put it on McConkey, but let's just say McConkey agar. And then it turned pink, so it is able to metabolize lactose. that's something you could say about that organism in that case. So that's just an example. So that's a differential way. You can also use selective media if we want to help isolate. So we might break it down to thinking that it's an enteric, or, usually it's stuff that would be found in the gut. Enteric bacteria. So you might go it on EMB, agar, like we're going to be using in the lab, to try to break it down to the enterics. It's McConkey because it has the bile salts in it. So whatever. And then we have blood culture. So you would have anaerobic and aerobic. Draw your aerobics first, always, to draw any air out of the line, and then draw your anaerobic after that. Okay. Biochemical tests that we'll be using, we're going to be doing a ton of these in the lab. They're coming up quick. So these are basically different kinds of media that have, to be restricted media that have some sort of additive into it. And can this organism live off of this additive? Can it metabolize it? And then based on color change in that media, yes or no, it does metabolize it, or it makes this kind of product, or it makes that kind of product. And that can tell us a lot about the organism. They have these strips nowadays that they show in the actual lecture, but they look like this, and they have these little wells in them. And each well represents a different test. So one of them might test for indole, and one of them might test for glucose usage. I promise that this makes sense when I type it, or when I draw it, but whatever. So each well, and there'd be like 20 wells or something. And then based on whether or not you saw a color change, or it could change from whatever color to whatever color, there's a key that you can compare it to. And if it's positive or negative, it gives you a number result, and you plug that number into a key essentially, and it tells you what organism it is. It is exactly the same thing as what we're going to be doing in the lab with our dichotomous key, except on when you use the strips, it's on a larger scale, and you have more choices than just the few bacteria that we are using in our dichotomous key. So dichotomous key might start off, you might have, whether it's gram positive or gram negative, you might start off with gram negative, and then whether or not, it can ferment glucose. Does it ferment glucose or not? And then if it does ferment glucose, I don't know, like, can it use starch? I just want to draw this out just a little bit so you can get an idea, or not. And if it can't use starch, let's see, good one, catalase. Does it possess the enzyme catalase, which protects against enzyme, oxygen, toxic oxygen molecules, right? It helps break down hydrogen peroxide into water and oxygen. So, and if it has, whatever, and we could leave it even like that. So you would just follow it down based on whatever your results were, and you can break it off more with more tests and whatever, and that might help you distinguish between what organisms what. This does that, but a computer system has that set up for you and already has that figured out for you. Okay. PCR, that's using genetics. So we're essentially going to be taking DNA from this organism. So we'll treat it in a certain way from your pathogen. And then we will, after we've broken the cells up and so we can access the actual DNA inside the cells, because inside the cells, we can pop it into a PCR machine. Now, I've told you this before, please do not forget it. PCR machines are not magic. You don't just pop DNA into it and they give you an answer. You have to have the appropriate primers to check for the thing you think it is. You have to have a guess. So, you run one PCR run to check for E. coli. Oh, it was negative. Maybe you run another PCR run for salmonella. And some machines might be able to run all of these at once and whatever, but it's not like you get the sample from the patient and you just pop it into a PCR machine and it's like, oh, Ebola. It's not how it works. You have to have the resources to test for that organism already available. It has to exist already. It's not magic. Okay. Antimicrobial sensitivity, that can actually be used for identification as well as trying to figure out what antibiotic to treat your patient with. We know about treating the patient and how you might want to use the Kirby-Bauer method to determine what antibiotic, your patient's organism is most susceptible to. But some bacteria might be more sensitive than others, and you might want to know if it's MRSA versus a typical staph, or a worse form of staph than MRSA, more resistant than even that, and all of that. So a Kirby-Bauer method is a way to help you differentiate that. So that, remember, is going to be putting bacteria all over a plate, and then placing antibiotic discs onto the agar. And then if based on how wide the zone of inhibition or the clearing around the disc is after it's grown, you can measure that and essentially determine how resistant, which means you know, it wouldn't be killed as well, or how susceptible it is based on that. Okay. Moving on. So immunologic tests that we can have. This includes things like ELISA, but our immunologic tests, some of them might be more specific, which means that we would have no false positives associated with that, because they're only going to interact with the positive and not with just random molecule, versus sensitive, which means that we will detect every positive, but you might sometimes have a false positive, but you'll definitely catch the positives. They can be a combination. If you want your thing to be as specific and as sensitive as possible, which everybody does, but there's only so much you can control with that. So that's the idea with the difference between sensitivity and specificity. In vitro serum testing. So this is taking serum out of a patient. In vitro is outside of the body. So we've taken a blood sample, we've spun down the tube, we've let it clot, we've spun down the tube, and we've taken the serum off. And now we can use this to do all sorts of testing. So immunofluorescence, which is literally using fluorescent marked antibodies to look at the presence of something or not. And if it glows, it's there, and if it doesn't, it's not. And then we had ELISA, which we did in lab, enzyme linked immunosorbent assay, where we would have, you know, if we're looking at the direct, we're looking for the presence of the antigen. If we are doing the indirect, we're looking for the presence of, does the patient have the antibody? Okay. So those are different ways that we can approach those tests. And I'm not going to go into too much detail about that because I feel like we've talked that to death, really. But that's applies if you don't know what that is and how that works by now. You should really go back into the textbook and review that. Because, yeah, remember, indirect, we're going to be, I don't know how to make it easier to remember. Only way I remember it is that if you were doing a sandwich, ELISA, where you have literally, you have stuck to the bottom of your well, you know, an antibody. So yeah, with these tests, you have to have a known. You either have to know the antibody or you have to know the antigen and what you're looking for. So in this case, with the direct, put a D, for the direct one, you have an antibody that you know, and then that's stuck to the bottom of the well, and then you put the patient's serum on there, and it has, I don't know, tuberculosis or whatever, and it comes in and it sticks onto that antibody you put in there. You do a rinse, then you add the same antibody, basically the same antibody, but this one is linked to an enzyme that whenever, you know, this is in the well, by the way, whenever you add the colorimetric substrate, which is a fancy way of the thing that turns a color in the presence of the enzyme. If this bound to this, then we have a color change and it's positive. That's what it means. So yeah, so this is the sandwich methods because it's on an antibody on each side. And that, I don't know why, but that's always, I always think when I think of the sandwich method, the direct method, but I don't know, I don't have like a way to help you remember that. Just trying to think of if there's a better way. I'll try to think about it. If I think of something that'll work, I'll let you guys know. But all right, Western blot. If you get a positive ELISA, ELISAs are they're sensitive, but they're not necessarily specific. So they might have false positives. That antibody interaction, it can be a little bit sketch, especially if you're not rinsing the wells well. So, so Western blot. So if you get a positive in ELISA, you might want to do a Western blot to confirm because it is very, very sensitive and specific, but expensive. So that's the only reason why we don't do them all the time. So we use these to confirm. These are confirmatory. Then we have, right, we already talked about this. Yeah, so indirect, that is the antibody present. Direct is the antigen present. I mean, think of it this way, I guess. If you're going to be making a sandwich, what's going to fit better in the middle? A weirdo antibody that's all Y-shaped, or an antigen? An antigen could, you know, be a nice little black guy that could sit in the middle. I don't know, that looks better to me. So the sandwich method, we're looking for the antigen, and that's the direct. We can also identify things by bacteria anyways, by phage typing, because bacteriophages, every bacteriophage every species of bacteriophage only infects one species of bacteria. So they don't cross over between species. So if it infects, you know, if you're trying to determine what something is, you could plate it out on a plate with, you know, a phage that only infects salmonella. And if you see little like clearings in there, that means that that's a sign of phage infected a cell and cleared out that area and multiplied. So those are called plaques. And that's would be an example of using phage typing to confirm the identity of something. So it only infects, you know, salmonella enterica, and that patient might have salmonella enterica if you have plaques. So, all right, that is it for chapter whatever this is, 17. So this is the end of unit 3 if we had units in the summer, but I did not split it up that way. So we're coming up on the second part. So that's going to be the diseases. So it should get a little bit more interesting for you guys.